Hello dear students, in this video we will study about Harshey and Chase experiment to prove DNA as a genetic material. Though through transforming principle also we got an idea about genetic material, but here Harshey and Chase experimentally proved that DNA as a genetic material. So in the year 1952, two scientists came, Alfred Harshey and Martha Chase, and they proved DNA as a genetic material by using bacteriophage as a genetic material. Now the question arises, what is bacteriophage? Student, bacteriophages are the viruses that infect bacteria. They are composed of protein and DNA. After that, they both started doing their experiment. They prepared a culture plate with radioactive phosphorus and allowed bacteriophages to grow into it. They multiplied with the passage of time, and these radioactive phosphorus got incorporated into the DNA of the bacteriophage. Now the question arises that why these radioactive phosphorus got incorporated into the DNA of bacteriophages only and not to the protein part. Because student, if you remember, I have told you that DNA is composed of nucleotide and nucleotide is composed of phosphorus. So wherever phosphorus is there, it will incorporate it into that part only. Clear? After that, again they prepared one culture plate, and now they took radioactive sulfur in spite of radioactive phosphorus. Here also they allowed bacteriophages to grow into it, and the result they found that radioactive sulfur got incorporated into the protein part of the bacteriophage. Clear? After that, student, they started doing their experiment. Now they took bacteriophage whose protein part was radioactive. After that, they allowed the infection of those bacteriophage with the bacteria. And during this process, transduction occur. What is this transduction? It is the process of transfer of genetic material from virus to bacteria. And this is the principle on which your Harshey and Chase experiment is based. After that, your second step is blending. First step is infection, second step is blending. What happened in this? The genetic material of virus as well as bacteria, they got blend. Third step is your centrifugation. Here what happened? With a huge agitation, these protein and bacterial part, they got separated. Heavier part will be at the bottom and lighter part would be on the top. Heavier part is known as pellet and lighter part is known as supernatant. So your lighter part is protein and heavier part is bacteria, and they both observed that there was no radioactivity in the bacteria part. So they concluded that protein is not a genetic material, because if protein would be, then radioactivity must be there in your bacteria. After that, they took another bacteriophage. Now this time they took that bacteriophage whose DNA was radioactive. Again they did same. First step infection, second step blending, third step centrifugation, and now they observed that radioactivity was there in the bacteria, and they concluded that DNA is the genetic material, because here they have taken radioactive DNA. So if radioactivity was found in the bacteria, it means that DNA has entered and it is only the genetic material. Clear? Now let's give you one challenge, whether you have understood the concept of not. Okay, so Harshey and Chase experiment was based on the principle, you have to tell me, okay, transformation, transduction, translation, or transcription. Just think over it and you have to answer me. Clear? Okay, students, I'm waiting for your answer. That's all from my side. Thank you and have a wonderful day ahead. Thank you so much.